Hello, and welcome to Bio 212, Anatomy and Physiology 2. We are working on Chapter 29, which is Development and Inheritance. And in this last portion, we're going to discuss the inheritance properties of what happened when a spermatocyte actually plus an ovulated secondary oocyte, okay, formed a zygote. Z stands for zygote, right? And what I need you to understand is that this is a haploid structure, and this is a haploid structure, and this now makes this a diploid structure. I'm going to just put dip here to make sure you don't get confused. Okay? Now, what that also means is that you have 23 chromosomes here, plus 23 chromosomes here. And now you actually have 46 chromosomes here, but the way it's usually referred to is 23 pair. 2, 3 pair. Right? And that's what we're going to be discussing. That 23 pair of chromosomes. And if I were to draw, let's go, this is one chromosome from, say, dad. Let's change color here. And this is going to be one chromosome from, say, mom. They're going to be attached, okay, by this structure here, so that literally there's no getting away from what's about to happen. What you need to understand is that there are going to be a series of genes on each of these 23 pair that have specific responsibilities inside of the human body. And the way to remember that, of course, is the central dogma of biology is DNA gives rise to an information intermediate that is called RNA. And that RNA holds the instructions so that we can assemble the amino acids correctly to give us a protein. Okay, and if you need to review that, it's chapter 3 inside your textbook, inside AMP1. And if you have questions about it, don't ever be afraid to ask. All right. Okay, fantastic. So that's the idea. So we have 23,000 of 25, 23 to 25, depending on whose numbers you're using, right? It seems like a big range, but there's a reason for that. You have all these different genes, and they have, let's put one here, and I'll put another one over here. These would be referred to as alleles of the same gene. So let's say gene for hemoglobin A over here for dad, and hemoglobin over here for mom, will encode, generally speaking, the same type of information, but there might actually be mutations within it. So there's something different about one or the other of these genes. And the significance here is it's an if. The entire portion of the inheritance section of your textbook is if something has happened, then you should be able to predict what you're looking for. So a mutation is a permanent heritable change in an allele that produces a variant. So let's put a little asterisk over here, and let's just say that maybe there's a mutation that confers some form of a disease state, which is what usually people are looking for to make this all make sense. Could also perform some sort of advantageous selection, but we don't tend to talk about that stuff in this class. Okay? So, and if this is the gene that's expressed, meaning DNA is transcribed into RNA, and the RNA is then translated into protein, well, then you have to think about what the consequence of transcribing that gene is. Okay. The first example that your textbook uses is fetal ketonuria. So I got to get rid of the cartoons first. Wham. Hopefully those are helping. I don't get feedback from you guys, so I have no idea. Okay, and there are a series of hereditary traits inside humans that we could go over inside lab. We should or could have gone over this PTC taster, which is your ability to taste something. And if you don't have it, it means you're a non-taster, which means that is the recessive allele that's being expressed inside of you. But what we did cover is instead, how do you actually assess if something can be expressed And your textbook, again, uses a disease state to do this. Because I don't know, maybe we think diseases are more important to study. And what you can see here is you have homologous chromosomes that could be used to fertilize a fetus, to create a zygote, and homologous chromosomes from a mother who's heterozygous as well. Okay, and what we mean by heterozygous here, of course, is you actually have big P, small p, big P, small p. If mom was homozygous dominant, she would have been, well, let's see if I can do this here. She would have been large p, right? That'd be homozygous dominant. If she was homozygous recessive, she'd be small p. Okay? And that is just paying attention to nomenclature. So you set up a Punnett square, and you'll notice that the mechanism or the repeated way they do it inside your textbook is they put mom on top, dad to the side here, and then they just sort of do the cross for you. Large P ends up in this quadrant here. Large P, small p ends up in this quadrant here. Large P, small p ends up in this quadrant here. And then small p end up here. And that means that inside the boxes, you're recording the possible genotypes. And you'll notice that there's always going to be possibly 4 because you have 2 possibilities here by 2 possibilities there, mean you have 4 possibilities here. How we interpret it, though, is the work. Because genotypes give rise to possible phenotypes, meaning possible genotypes could give you possible phenotypes, and that's the analysis piece. And what you're looking at here is if you wrote out all of the possible consequences of a cross, you would see that there's one homozygous dominant or one large P. You could see that there are 2 large P, small p's here, and these are heterozygous dominant, where the dominant allele, if present, will be the allele that is expressed. And then you have one homozygous recessive. That's the small p that you see here, and therefore that is the one individual that expresses the mutation that is the inability to function properly, and we call that phenylketonuria. You'll notice I'm not even telling you what the disease is. It's more important that you understand how to set up a cross and analyze the cross. Okay. Let's get out of the cartoon. Let's go over here. So how can these things happen? How can you actually end up with, just mistakes? Let's just call them mistakes. Your textbook calls them mutations, and I think that scares people, but they're mistakes. Alleles that code for normal traits are not always dominant. Okay, if you're looking at, say, Huntington's disease caused by a dominant allele, both homozygous dominant and heterozygous dominant end up with Huntington's disease. It appears to be, in this case, the function of the protein is sufficient to cause the phenotype that you see. Whoops, let's go back to this. Imagine, if you will, when we're going through our normal mitotic events, if you pull the number of chromosomes, so you actually have the wrong number of chromosomes inside of the resulting sister or daughter cell, right? You now actually have missing a chromosome, or you have an additional chromosome. Mother Nature doesn't like to lose chromosomes, and Mother Nature barely stands with getting additional chromosomes. Okay, and what I mean by that is, yes, Down syndrome individuals expressing extra chromosomes of 23 are alive, and they have extra chromosomes, but it comes at a consequence. And thinking about it, just here off the top of my head, I don't actually know what the mechanism is. I remember reading once that it's a dosage problem, meaning you have extra proteins that are being made as a result of transcription, translation, and the ultimate production of proteins. But I'm not sure anybody has actually worked all of that out yet. So you actually can have this really simple system that we set up earlier, right, where we actually have a large P and a small p when we're looking at phenylketonuria. This collectively is what we would call a dominant recessive inheritance system, where you actually have the simple dominant recessive. You can actually have incomplete dominance, where neither allele is dominant over the other. Okay, you could express either one and neither is not dominant. You could have heterozygous, which are intermediate phenotypes, And the disease state we're going to use to examine this is sickle cell. And I know we looked at it in class. And what I mean by that is, let's take a look here. Get rid of the cartoon. Go here. And HB is hemoglobin. And you can actually have HbA, which is the normal hemoglobin allele. HbS is the sickle cell disease. And you can actually have HbA, HbS, where you actually have minor problems. There are still some intermediate difficulties with red blood cell formation, oxygen transport, stuff like that. However, if we go like this is the individual down here that has the full-blown change. So if this is a normal red blood cell in terms of surface area, notice it's a biconcave disc. This should be in red, right? Okay. And remember, the sickle cell in its sort of harshest form, right? It becomes these really sticky structures. And when you're trying to get through, let's do this. When you're trying to get through, say, a circulatory structure, put a capillary bed in here for you. I'll put that there. If you're trying to move a red blood cell through a capillary bed, well, if it's sticking, let's just say it creates a bottleneck here, right? That means everything down here isn't going to function properly. And that's actually where the danger comes from with expressing the recessive homozygous phenotype here. Okay? Cool. Let's get rid of that. Again, don't try to memorize this. Reason your way through it. You have an A here, an S there. The only thing that's different here is we call it hemoglobin, right? So it's actually how you can see the consequence. That could be a P right there. Same idea. Okay. Multiple allele inheritance. Some genes have more than 2 alleles, right? And I think what they're trying to say here is that if we're going to make, say, antigens that are expressed on the surface of a red blood cell, where both A and B are codominant, what you can actually see is A, or A with a small I here. You end up with an A-type phenotype. You have B with a small I. You have a B. Large A, large B, you have AB. And the isotype with the small isotype here gives you the O. That's where the O's actually come from. So it's the lacking of the actual glycoprotein on the surface of the cell. So why do we care about that? Well, remember, it's not about you, it's about the offspring. So you could actually end up with an O-type offspring even if both mom and dad are A. And again, like I said, don't try to memorize these things. Think about what would actually have to be done to actually get, if you have an AB, it could be a B, an AB, or an O. If you're an OO, it's always going to be O. In fact, believe it or not, that's the way that I actually remember all of this. If you can remember that, if we go back here, the only way you can end up with an O phenotype is if you're lacking the AB markers. The rest of it starts to make sense. Now, complex inheritance, the textbook example, of course, is, you actually have polygenic inheritance where most traits are not controlled by one gene. And I think I've sort of tried to stress this to you. Remember when I said we're putting together Punnett squares, it's the application of the idea. And if you try to do it for 23,000 genes, you can see how complex it would get. Now, that's your skin color, your hair color, your height, your metabolic weight. You can actually see what this looks like if we look at, say, the color of skin when you're looking at, let's say there are 3 different alleles for mom and dad, where you actually have, I'm just saying they're mom and dad. So let's see, what did they put up on top here? So possible ova here, possible spermatocytes here, and you can see the gradation in pigmentation. Okay, which makes sense. Very light, you end up with a very light pigment over here. Very dark, and very dark, you end up with the darkest pigment on this corner here. And just go 6, 1, 2, 3, 4, 5, 6, 7, 8. Okay, how do you end up with 8 permutations here? How do you end up with 8 permutations this way? That's the possibility of 64 possible gene dosages to give you the skin pigment color. The next and/or last topic that we're moving into here is, well, these are the autosomes. 1 through 22 are 22 pair of autosomal chromosomes, and you can see 1, 1, 2, 2, 3, 3, 4, 4, 5, 5, 6, 6. And these pairs of chromosomes, okay, are arranged by their actual size. So the histologist, when they actually get a blood smear, these don't look like this at all. What you do is you take pictures of them, you rearrange them based on the chromatid arm length, right? That's actually how it's done. And you can see there are 22 pair of autosomes, both in males and females. The differing category here, of course, this is a male because this is a Y chromosome. So using the textbook sex determination pathway, look, this is just a Punnett square where the only real difference are the genes that are expressed on XY versus XX. Okay, and if we use the SRY gene, oh look, there it is. If we use the SRY gene as an example, it's only going to be found on the Y chromosome. So that means the only place you're ever going to see Mullerian inhibiting substance is going to be inside the male. So that would be a male-linked expression of a gene. Okay? If we think about, say, colorblindness, right? Red-green colorblindness, which is, I think they've been teaching since, I don't know, 3rd grade or something like that. You're looking at, here's the potential genotype, and you'll notice that it's X, large C is a normal female. X, large C, X, small c is a normal female, meaning her retina are functioning properly, but she's a carrier. A colorblind female is X small c, X small c, so that would mean this person would actually have red-green colorblindness. Okay, this woman would have X-green colorblindness. Now when you're looking down here, you're looking at the males, you'll notice that it's not carried on Y at all, ever, so therefore the only time you actually have a colorblind male is if the X chromosome is carrying the recessive form. Okay? That makes sense? Cool, because notice what we're saying here is the only time they actually get that is if they inherit it from their mother. Cool. And this is what the Punnett square would look like to that. Okay? Here are the males along the side here. Here are the females along the side there. You break down the 4 possibilities here. There's a 25% chance if mom is a recessive carrier and dad is normal, meaning a dominant red, green, blue color vision capable. There's still a 25% chance if mom is a carrier that you end up with a colorblind male. Okay, so let's do the overarching sort of summary for chapter 29. So chapter 29 begins with fertilization, and we go into the mechanics, namely the how do we actually build an inner cell mass that can be supported by a placenta and create 3 distinct tissue types, ectoderm, endoderm, and mesoderm, so that you build a functioning tube inside of a tube that has a nervous system that has the 5 characteristics of all living organisms, that at birth, because of changes in concentration of estrogen, progesterone, okay, and cortisol, you now actually can have a living organism outside of the mother. Mother's engineered to still allow for the survival of the organism. So now we think about the required changes for the fetus, meaning we close the portal system inside of the fetus. We have functioning lungs now, but also the inhibition of prolactin production inside mom is now released. So you release the inhibition, and now prolactin can drive the production of milk for as long as milk is needed. Okay? From there, what we did is your textbook switches to this idea that, well, every single living organism is the product of both maternal and paternal genes. And we went through a series of different types of inheritance that now we can simplify to this Punnett square system of analysis where you can look at the potential genotypes that would then be seen as a phenotype inside of that offspring. Remember, we went through general dominant recessive, then we went through sort of incomplete dominant recessive, then we went to sex determination, okay, as well as multi-allele or complex inheritance patterns. For the purpose of this class, make sure you understand the definitions of all of the inheritance patterns, but if I'm you, I'm spending more time thinking about the mechanics. Okay, thank you very much. This is a really good place for us to stop, and have a great day.